to all of you. Welcome to another session of NEET Bytes by Meritnation. And this is me again, Kausia, who will be conducting another beautiful session about some conceptual question on the same topic, which is cell, as we have done in the previous part. And I told you that because this is a big topic, it involves this discussion on different organelles. Hence, we will be conducting in two parts. Okay, so I welcome you once again. Welcome, Kavita. Hi, Shehroz. Good evening to all of you. Hi, Smriti. I'm sure you guys have come prepared because just like a previous session, we will be doing it in the quiz manner. Okay, in the rapid fire round that we did in previous class. You guys remember? Right. Okay, so we will be having the same thing going on back again here. And it will be fun, right? So you'll get to know how many questions you are doing right and how much time is it taking for you to understand a question, answer the same. Yes, of course. So Shreya, welcome. Vishnu, good evening. And Harsh, good evening. Okay. Yes, of course, we are going to have quiz today as well, Meghna. Today as well, we'll have the rapid fire round for each question. So those of you who were not present in previous session, those of you who are joining from this session, let me just tell this once again. So I'll be projecting the question, right? And for each question, I shall give 1 minute exact. Okay. In that 1 minute, you are required to read the question, provide the answer for the same, right? And one person will answer only once, right? One person will answer only once. And after the minute is over, you will not answer then. Your answer will not be counted after the minute. Okay, and likewise, we'll continue for all the questions that we have today. There are 10 questions. We'll continue for all of them. After that, at the end of the session, I will be announcing the top rankers. Okay, I will be announcing the names of those people who have acquired rank 1, rank 2, and rank 3. Right, exactly. So this is what we are going to do today. Fine. So shall we start now? Let's get started without much ado. Okay, we'll be looking into more of the cell organelles. I'm sure that you have come prepared. Very fine. Okay, so here comes your first question, right? Okay, so very good. Starting with first question. This is question number 1. Your time has started. In this question, you are required to tell me the correctly matched option. Okay, here are the names of the parts of the cells. You are seeing the plasma membrane, the mitochondria, the chloroplast, and the Golgi. And some discussion and some of their properties are written right in front of them. You have to read them and you have to tell me the only correct option from the 4. Right? So about plasma membrane, do you think it is made up of cellulose or it is made up of chitin? Do you think it is absent in some cells? Bacterial-like elements, mitochondria are like bacteria, and they have inner membranes forming sac-like structure. They bear chlorophyll. Do you think this is right? Chloroplast are bacteria-like element. Inner membrane is highly folded. Is that true for chloroplast? Something that you know. Golgi is a stack of flattened apparatus vesicle. Do you think this is right? So quickly put up your answers. Your 1 minute is over. Okay, now we are going to discuss the correct option here. So as I can see, many of you have correctly guessed the right option and D is the right option. Okay, for you know that first option doesn't talk about plasma membrane. It necessarily talks about the cell wall. Okay, because it is cell wall that you find made up of cellulose and chitin. For plant kingdom, it is cellulose. For fungi, it is chitin. Okay, and it is absent in some cells. So yeah, if we talk about animal kingdom, we do not find cell wall there. So this is not for plasma membrane for that matter. It is a cell wall. Okay, Ankit, very good, Ankit. If it is confidence, I'm highly impressed. Okay, if we talk about mitochondria, then of course they are something like bacteria, but they do not have chlorophyll. That's not true for them. Hence, this is not correct. Similarly, if we talk about chloroplast, we do not find folded inner membranes. The inner membrane of chloroplast is continuous like an outer membrane. However, you find a third system of membrane called thylakoids there. Right. And you know that thylakoids are arranged in the form of a stack-like structure, forming the granum for that matter. So only fourth option that talks about Golgi, it's a stack of flattened apparatus vesicles, something if I happen to draw, okay, that comes out to be like this. Okay, could be poorly drawn, but I'm sure you understand that. So this is the only correct option. Okay, so that's fine. I think this was an easy question. So are you guys ready for the next one? Any sort of issue in that? I'm sure that's cleared. Let's move on to the next question. What do you guys suggest? Very fine. Moving on to next question. Right. Time has started and the question is asking you about something called as polysome. You have to think, what is polysome? How do you form it? Right. Option A says that ribosomes attach to each other in a linear arrangement. Ribosomes are attached to each other. This is what you have to think. Several ribosomes are attached to a single mRNA. Is that true? Many ribosomes attach to a strand of endoplasmic reticulum. Does that fit the polysome description? A ribosome with several subunits. Do you think this is right? So basically, all the options are talking about ribosome. So this is something that you get an idea here that we are necessarily talking about ribosomes. But in what condition? Polysome will result in what situation? Okay, so I can see many people answering me. I'm going to reveal the right option within few seconds. Get ready, embrace yourself. Right, so your minute is over. And congratulations to all the people who have put up option B as the right answer. So, right, polysome is something that you find when to a single mRNA, let's say this is a single mRNA molecule. Okay, on that, there will be arrangement of many ribosomes side by side. Okay, many ribosomes will be attached in this particular fashion as I am trying to draw on the board right in front of you. Okay, this is what something you find. This is polysome. Okay, they are not attached together in the endoplasmic reticulum. You don't call them polysome in that fashion. Okay, and those of you who are in class 12th right now, and those of you who've studied about your molecular basis of inheritance chapter, you better understand that polysome is something which is a common site in prokaryotic cells. Am I right, people? Am I right? You find that very frequently in prokaryotic cells because there the transcription is not separated from translation. Okay, exactly. So you can relate from that learning you have made in your syllabus there. Okay, Adil, it is polysome. We call it normally as polysome. So basically, if you break up polysome, you will understand it like polyribosome. So right, that is for understanding basis, it's correct. But we use the term polysome here. Okay, Adil. So that's correct. If you are breaking polysome, you will get polyribosome. That's fine. But we do not write the term as polyribosome. We better write the term as polysome. Okay, understandable, everyone? So you find a single mRNA molecule is being attended by many ribosomes together, and then they are performing their task of doing the translation. You'll find the polypeptide chain coming out from all of these. Okay, so those of you who are in 11th class this year, you will understand this more clearly in your class 12th. But then you have a basic idea about polysome this year. Okay. Right. So I guess this is clear to all of you. We'll move on to next question, everyone. Right? Okay. Here comes next question. And your time starts now. The question is asking you about the important sites for the formation of glycoproteins and glycolipids. So it talks about the process where sugar moiety, okay, the sugar moiety is added onto the proteins, it is added onto the lipids. This is one and very easy question in the entire universe, I tell you, right? This is something that you study when you talk about that particular organelle. So take your time and understand, and then I will tell you a little bit modification in the question. If the question would have been like that, then how your answer could have changed, right? I'll tell you that. So my intention of projecting this question is to tell you the modification basically. So I can see many of you people are guessing the answers basically with confidence. I'll not say you are guessing the answers. You do have the confidence for the same. So your minute is over, right? And the correct answer is option C, as you have guessed. Many of you have guessed is correct. Okay, now one thing I want to tell you, if in the question, listen this very carefully, if in the question there would have been a term called N-linked glycosylation of protein, suppose if they would have asked you this thing, okay, because glycosylation basically is the process of addition of sugar moiety there, okay, then how your answer would have changed? So would you have gone for endoplasmic reticulum, or would you have gone for the Golgi bodies? Can I have the answers once more? Like, this is simply, if you understand what I'm trying to say, that suppose if in the question you have had N-linked glycosylation, if the question had asked you like that, what would you have chosen in this case? Quickly, what would you have chosen in this case? Can I have a few more answers once again? Right, so I'm getting answer from Anuj Pandey, very good. Ayush Gupta, Avreet Aditya, perfect. Then you would have gone for endoplasmic reticulum. Very fine. Because in Golgi bodies, the O-linked glycosylation occurs. Okay. In Golgi bodies, you find O-linked glycosylation occurring. Perfect. So you know this thing. Very good. Keep it up. Keep it up, people. Very fine. So now let's move on to next question. What do you think? Perfect. So this thing is clearer to everyone. Okay. Moving on. All right. All right. So next question. Okay. So your time starts now, everyone. Time starts now. Please tell me, where do you find DNA? Okay. Please tell me, where do you find DNA? Okay. So you find it in chromosomes and dictyosomes. You find it in chloroplast and lysosomes, in mitochondria and chloroplast, in mitochondria and endoplasmic reticulum. Basically, in this sense, we are talking about extra nuclear DNA. We are talking about this DNA, which is not present in the nucleus, and this is also known as cytoplasmic DNA. This is another term that we use for the same, cytoplasmic DNA. So in cytoplasm, essentially you have organelles present, right? So in which organelles will you come across the DNA molecule? Right, so your time is over and I have gotten many answers already. So a big congratulations to all the people who have given me option C as the answer. That's right. Mitochondria and chloroplast, somebody is writing. Aditya, very good. They are considered to be semi-autonomous for that particular reason. Okay, we are considering them semi-autonomous because they do have their own DNA, which is capable of forming some proteins for them, not all. Okay, that is why they are semi-autonomous. If the DNA is coding for all of the proteins, then they would have become autonomous. But no, they are under the under the control of the nucleus. Okay. RER or SER in the previous answer. Ankit, we are talking with reference to the proteins. Okay, and you know that proteins are coming right from the ribosome and going to the rough endoplasmic reticulum because ribosomes are attached on the endoplasmic reticulum. Understand? So the answer would be more precisely as rough endoplasmic reticulum. Okay, Ankit, I think I have taken up your doubt. And here people understand that your mitochondria and chloroplast are semi-autonomous. That is why they have their own DNA, which is also termed as cytoplasmic DNA or extranuclear DNA. Okay, so this is a right option. Big congratulations to all those who have given me the answer. Okay, so I guess up till now, everything is moving good. You are able to guess and confidently answer many options correctly. Okay. All right. So I will answer this question of yours, Singham. Okay. And let me just go to the next question. And I'll explain to you why do they have 70S ribosomes. Okay. So moving on, next question. This is an assertion reason question. All right. This is an assertion reason question. Let's get started. Your time has begun. And let's go to the assertion quickly. Let's try to read the assertion. Says that the true nucleus is generally absent in Escherichia coli and other prokaryotes. So do you think this is right? Assertion is telling you a right thing, that Escherichia coli, which is essentially a U bacteria, doesn't have a true nucleus. Okay. And the reason they are providing for the same is that an undifferentiated, unorganized fibrillar nucleus without any limiting membrane is observed in prokaryotic cells. Okay, so you do find a kind of a nucleus, but that doesn't fit into the definition of nucleus because it is undifferentiated, it is unorganized, it's not having some boundary structure. Hence, we do not call it as nucleus, but we call it by some other name. You have to come up with the options A, B, C, D depending on both the statements' validity. Okay, whether they are valid, and after that, even if they are valid, are they complementing each other? This is what you have to think. So your minute is over, right? And a big congratulations again to those people who have given option A as the right answer perfectly. We know that prokaryotes are basically prokaryotes for the reason that they do not have a defined nucleus present. Okay, yes, they do not have a defined nucleus present for the reason that there is no limiting membrane around the nuclear material. Understand, their DNA is not surrounded by a particular membrane. Hence, we don't call it as nucleus. What is the term that we use there? Can anyone suggest me the term? Can you suggest me the particular peculiar term we are using to refer to the genetic material of prokaryotic cells? Anybody? I'm sure you know the answer. Okay. This is question within question. I'm sure you know the answer. I've gotten an answer from Singham, from Varsha, and Aditya. Very good. That's your nucleoid. Perfect. Perfect. We call it as nucleoid. So this is a feature of your prokaryotic cells. And Singham, coming to your question now, you were asking why mitochondria and chloroplast have 70S type of ribosome. So basically understand that we believe the appearance of mitochondria and chloroplast inside the eukaryotic cell has occurred due to some insertion of free living bacteria. Okay, so mitochondria and chloroplast both are believed to be free living before they got engulfed by a eukaryotic cell. Okay, and there in the course of evolution, they lost their autonomy. Okay, they lost their autonomy to the nucleus of the eukaryotic cell. Right, but essentially they do have the prokaryotic structure. That is why you find 70S ribosomes there, and also you will find that their DNA is not surrounded by a nuclear membrane. They do have DNA, okay, but that is not surrounded by a nuclear membrane. They also, for that matter, have nucleoid. Okay, just like that. So you have to understand that within a eukaryotic system, you are having a prokaryotic system. Okay, the prokaryotic system is retained within the eukaryotic system when you talk about mitochondria and chloroplast. Okay, so I guess I have given answer to your question, boy. And this question is also very good. You've corrected, you've come up with correct answers. So let's move on to the next question. Everyone, shall we move on to the next question? Are you guys ready for the next projection? Cool. Moving on. Here comes next question asking about mitochondria and chloroplasts once again. Okay, here you have two options present in front of you. Your time has started. You are supposed to tell me which one is true, which one is false. Perfect. First option says that, first statement says that they are semi-autonomous organelles. Well, I'm sure that you know the answer. This time you know what they are. And the second statement is saying that they are formed by the division of pre-existing organelles. Okay, and they contain DNA. Okay, but they lack protein synthesizing machinery. Okay, that they do not have something which is synthesizing the proteins. What do you think is correct? What do you think? That statement 1 is correct, statement 2 is correct as well. Mitochondria and chloroplast cannot make their own proteins. They have to depend on the cytoplasmic proteins coming from the nuclear genes. Is that what you think? So let me have your answers quickly, all of you. As I can see, many people are answering with confidence. That's right. So your time is over. And those of you who have given option B are correct. A big congratulations to you. First statement is absolutely true. We know that. Okay, we've discussed about that. And just now I told you, just now before that I was referring to the query of Singham here, and I just told you that they do have 70s type of ribosomes present. And what are ribosomes? Ribosomes are nothing but protein synthesizing machinery, so they do have it. So you cannot say that they lack protein synthesizing machinery, for God's sake. They do possess. Hence, statement 2 comes out to be false. So another beautiful question, another easy question. Very good, very fine. So you are getting it right. Perfect. Moving on to the next question. Right? Shall we go on to next question? Everyone, what do you suggest? Shall we move on to the next question? Of course. Let's move. Next question. Okay. The time has started, and this question is about match the following. Here, you have column 1, which is giving you some structures, and on the opposite side, you have column 2, which is giving their description. So you've got thylakoids. Christy, it is, the spelling is some typo error. It is Christy here. Cisternae and chromatin. You have to come up with their perfect description. This is a fun question, right? Let's see how many of you are able to make it correct. This is Christy. Okay. So thylakoids, what they are? Where do you find them? Christy, what they are? Okay. Right. So I have scrolled down. Okay. I will give you some extra time here. Don't worry. Okay. So I'll add 10 seconds more. So you'll have 1 minute, 10 seconds to go to answer this question, because it does require some sort of match the following. Right. I have scrolled down. I have already scrolled down, I guess. This is perfectly fitting the screen. So your time is about to get over. Come up with the quick answer quickly. So I can find many confusions here. Time is up. Okay, let's try to solve it out. Okay, let's do it here. Thylakoids is something that you see in chloroplast. Okay, these are the membranes which are housing the pigments. Okay, they are carrying the pigments. So that is something you find in the chloroplast. So according to the column 2, if you read, the flat membranous sex and stroma of chloroplast makes the correct sense. Right? Makes the correct sense. Talking about Christy. Okay, now Christy is some finger-like structures, finger-like projections, right, that you find in the mitochondria, where if you guys remember in the previous class, I told you about the electron transport chain, right? And I told you that electron transport chain is nothing. It's just there are proteins present which are passing electrons to each other. That is where they are present, right? So Christy is something that is present in the mitochondria. They are infoldings in the mitochondria. Okay, talking about cisternae, of course, these are the disc-shaped structures present in the Golgi apparatus. They are basically making the Golgi apparatus. It is made up of cisternae. Okay, and then chromatin is the condensed structure of the DNA. Okay, it is the condensed structure of DNA. If you further condense the chromatin, you will get the chromosome, right? If you further go on condensation of the chromatin, you'll get the chromosome. So based on these matchings, we come up with B as the right answer. So those people who have given me option B as the correct answer, big congratulations, you got it right. Okay, you got it right. Exactly, Leshram. That's fine. Yes, Varsha, you are trying to say something? Okay, so this was another good question. It required some brainstorming. Now, I will move on to the next question. Okay, I'll move on to the next question. So, time starts now. This similar question I have brought in the previous session as well. Okay, this is asking you about the presence of some critical ion, but today you need to tell me for the unison of subunits of ribosome. If two subunits of ribosomes, okay, have to come together, which ion necessarily has to be present? Okay, this is another fun question, and the same type of question I brought in the previous session, but that was related to the assembly of microtubules. Remember, that was related to the assembly of microtubules. So I told you that there are certain divalent ions that you need, without which they will not assemble. Okay, so I'm getting many answers already. And let's see how many of you will come up with the right ones. So, few more seconds to go. Take your time. Those of you who haven't answered, do it now. Okay, we are about to reveal the correct option. Okay, so those people who are coming up with option C are right. You need to have the critical presentation of magnesium here so that your two subunits of ribosomes can remain combined together. Okay, and those people who are in class 12th right now, those who have already studied about the molecular inheritance of molecular basis of inheritance, you know that both these subunits need to come together in order to continue the process of translation, right? So you can relate it from there as well. Okay, very good, Paras, you got it right. Okay, so another easy question. Moving on to the next question now. Okay, time starts now. Ribosomes are produced in where? Which particular structure mentioned in the options is responsible for the synthesis of ribosomes? Is it nucleolus? Is it cytoplasm? Is it mitochondria? Is it Golgi body? Now, this is a very easy question, okay, because this is something that you very peculiarly you make it while making the diagram of the cell, okay? Exactly, you make it something like that. So Adil has started singing the song with like that. Very good, Adil, keep it up. So this is very easy, and take your time, and all the people who are not answering, please be a part, come up with the right option. Few seconds to go before I reveal the answer. Okay, so correct answer, your time is up. Correct answer is nucleolus. Okay, we all know that nucleolus is a dense structure that you find in the nucleus. Okay, and nucleolus, for that matter, is totally responsible for the formation of RNA and proteins that are going to be forming the ribosomes. Okay, yes, they are forming the ribosomes. And what exactly are ribosomes? Nothing. Ribosomes are ribonucleoproteins, isn't it? If you have to tell me the nature of ribosomes, you'll say that, ma'am, they are ribonucleoproteins, isn't it? Proteins are proteins. Ribonucleo basically we use for the RNA. So they are RNA plus proteins, and they are coming from the nucleolus. Okay, so that's right. That's right. I can see people are so happy singing the song with options. Okay, people are singing songs with option. I just hope what song will come with B option, C option. Keep it up. Keep it up. Innovation. Okay, moving on to the last question of the day. Last questions of the day. Question number 10 it is. Time has started, and let us do the question. Genes which are located on mitochondrial DNA, what type of inheritance are they showing? Okay, that a person will inherit mitochondrial DNA always from the maternal side. So they are showing maternal inheritance. They are always inherited from the male parent. Okay, that they are coming via sperm or they are coming via ovum. We can simplify it like that. They are coming via ovum, they're coming via sperm. Okay, or they are showing biparental inheritance, like they are coming from both. Okay, they're coming from both ovum and sperm. Is that what you think? And they are just not inherited at all. You have to make your own novel mitochondria. You do not get it from either of the parents. So, well, let me have the answers, and this is going to be a fun question. I can see. Okay, so your time is up. Let me reveal the answer. So, people understand one thing. Okay, ovum and sperms, both of them are responsible for contributing into genes, but not mitochondrial gene. Mitochondrial gene always comes from the ovum side, never from the sperm side. Sperm only contributes to the nuclear gene. Okay, it only contributes to the nuclear genes, whereas ovum is contributing into both. You are getting nuclear gene also from the ovum as well as cytoplasms, right? So mitochondrial gene is also coming from the ovum side. So it is always showing you maternal inheritance. Okay, so A is the correct answer. So all the mitochondria, okay, you get it from the ovum. Fine. All right. So this is the time for me to reveal the name of those people who have given me the correct answers. Just give me few seconds. All right, everyone. So exactly, Nishu, what I'm trying to say is that before the formation of zygote, okay, before the formation of zygote, you've got ovum and sperm present. Ovum is the cell which is coming with cytoplasm. Sperm doesn't come with cytoplasm. Okay, sperm is never contributing into the cytoplasm, right? And mitochondria essentially is present in cytoplasm. It's an organelle, right? So cytoplasmic contribution is made by the ovum. In that sense, mitochondria is also contributed by the ovum and not by the sperm. It shows maternal inheritance. It means that it comes from the mother side and not from the father side. Okay, so let me just have the names of the people, those people who have come up with the right answers. Right. If you want to ask something in that meanwhile, please let me know. If there is some question, let me know. If there is something that you want to discuss. Okay. Yeah, of course. Let's hope, Shreya, that I get your name. All right, people. So time for me to announce the name. Okay. All right. So just a few more thing. Okay, fine. Finally, I got your names. I know this is I'm taking so much time. I'm sorry for that. Okay. So in the first rank, everyone brace yourself. In the first rank, I have Aditi, Paras, and Smriti. All three of you have gotten 9 out of 10 marks. A big congratulations. Keep it up, people. Okay, 9 out of 10 marks. Very good. On the second rank, I have gotten Ayush, Dimple, Kavita, Leshram, Nishu, and Priya. Very good. So you have gotten 8 out of 10 marks. Okay, very good. On the third rank, I have gotten Aditya, Aman, Avreet, Meghna, Rahul, Shahroz, Singham, and Varsha. Congratulations, you've gotten 7 out of 10 points. Very good. So this is outstanding performance for that matter. Okay. And some of you, okay, I think the first ranker people have lost their marks in this question because you all thought it is showing biparental inheritance, isn't it? But this is a tricky question. All right. All right. So well done, every one of you. Well done. So I hope then I should take your, you will be studying more for your upcoming sessions because all the sessions will have the same pattern. We will be doing a rapid fire round like that. Okay, so I'll take your leave now. We will meet in the next session now with some more topics, more conceptual questions, more rapid rounds going on. Till then, all of you, be in good health. Take care of yourselves. Keep studying. Have a good life. Bye bye. I take your leave. Bye, everyone.